Chapter 4, Tissues. Maybe. There we go. So the tissue is the living fabric of the human body. The definition of a tissue is a group of cells similar in structure cooperating to perform some function. There's all manner of different tissues. Histology, that is the study of tissues. It's also a department in most hospitals. It's a great place to work. We break tissues down in the animal kingdom, of which humans are in the animal kingdom. We break tissues down into 4 different types: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissue, we say lines, covers, and protects, and is also glandular. Glandular as in provide secretions. Those secretions can be mucus, they can be hormones. Lines, covers, and protects. So the lining of the hollow cavities of the body, the stomach, the rest of the digestive system, the urinary tract are lined with epithelial tissue. Covers, some of the organs have epithelial tissue covering the outside of them. For instance, the pericardial sac of the heart, the pleural membranes of the lungs. And protects. Number 1 protection from epithelial tissue is the epidermis of our skin. So lines, covers, protects, and is secretory. Connective tissue is sort of a garbage can of all sorts of different things. The general definition of connective tissue is cells in a non-living background material. Cells in a non-living background material. So that background material can be solid, as in bone, or it can be liquid, as in the plasma of blood, and it can be anything in between. So cells in a non-living background material. Muscle tissue. Muscle tissue is one of the excitable types of tissues. Excitable because when properly excited, it can contract in response to a stimulus. That stimulus often comes from the nervous system, but it doesn't have to. It can come from other places. We have 3 types of muscle tissue. We have skeletal muscle, which is attached to the skeleton and the skin of the face. So we have facial expressions and we can move. Cardiac muscle tissue, found in the heart, contractile part of the heart, and smooth muscle, which lines is found in the hollow cavities of the body, for instance, to move food through the digestive system, or open and close a passageway like the entrance to our stomach or some parts of the circulatory system. Nervous tissue has 2 basic parts to it. Then the main player in nervous tissue is the neuron. And the neuron can initiate and propagate, pass on, an action potential, a nerve impulse. Now, the other type of nervous tissue is the supporting cast. Those are called glial cells, and there's a variety of those types, but they support the efforts of the nervous tissue. Also excitable. So here's some other examples. The nervous tissue obviously is found in the brain, spinal cord, and the nerves that come out of the spinal cord and go out to the body. Let's see. I think we got all this covered. Now, connective tissue, so not only is it bones and bloods like my example, but tendons obviously connect. Tendons connect muscle to bone, while ligaments connect bone to bone. And also connective tissue includes adipose tissue, otherwise known as fat. In order to view tissues, they need to be viewed under a microscope because cells are microscopic. and they need to be fixed. So that is fixed means preserved and so that it won't degrade. Then, since we're going to run it through a microscope, and we're going to need to pass either light or a beam of electrons through it, it needs to be in a very thin section. And then there's not much contrast at the molecular level, cellular level. So most of the time they're going to need to be stained with some type of stain. Sometimes you'd use methylene blue if you want to look at the nuclei of cells, or you might do a Wright stain. That's W-R-I-G-H-T, a name. Wright's is the name of a person who came up with scheme for staining and the ability to visualize blood. And there are others. Electron microscopy gives us a higher resolution. Light microscopy has a limitation of they say 1500 multiple magnification. I don't know about that. I've never seen that. The most I've ever used is 1,000 magnification. And when you get to that level, you're starting to get to the frequency of light, and it acts weird at that magnification. So to get any higher resolution or be able to see anything smaller, we use a beam of electrons, and that was discovered recently. Well, recently for me anyway. It was in my lifetime. And there's two types. You can use a beam of electrons through the sample, or you can coat it with some sort of heavy metal, oftentimes even gold, and bounce the electron beam off the outside of an object. Then one is transmission electron microscopy, TEM, sends the beam of electrons through the specimen, and scanning electron microscopy, SEM, bounces the beam of electrons off the object. And they're different instruments. The same electron microscope can do one or the other, but not both. And here gives us two comparisons. These are structures known as cilia. They're described as small and hair-like. They beat to move material across the surface of a tissue. And this picture on the right here is a scanning electron microscope that gives us the three-dimensional view, the outside of an object. The transmission electron micrograph gives us a high-resolution image of the inside, a two-dimensional view of the inside of an object. And if you look at this, you see, well, what are these things out here? We're taking a very thin slice. So it's possible, let's say we take the slice, say right here, that the object may go out of the field of view and then come back in. So let's say this one, it might wiggle its way out of the field of view and then come back in here, and then go back out and come back in here. And so particularly with cylindrically shaped structures or tubes, it can be difficult to interpret. If any of what I just said makes sense to you, then maybe you have a future doing this. And interpreting images is a lucrative field. That means it pays well. All right, so I'm going to stop right here and come back with a separate presentation on epithelial tissue.